Everyone, and I think we are running late of time, so without wasting the time, thank you, the chair. And I will briefly discuss the recent research which I am currently doing on the electropeptasensor. So there are three keywords: electronics, peptides, and sensor. And using these three integration, now we are developing a new biosensor that can be used to meet the challenges that we have in the unreachable biomarker-based diagnostics for any type of disease like seasonal diseases. We have influenza virus and dengue, chikungunya, malaria. So how we can develop a very affordable, low-cost devices that can be used by a common man in our society. So that is one of the target of this project. And I would like to highlight, because this project can only accomplished because of my dual affiliation both in India and Japan. So a little bit I would like to highlight that there was a need of that, because the technology has been developed in Japan and the real application which I realize can be done in India. So I think there is a good combination of these two countries. So basically I am working as a bioengineer. We started, I have two major applications. One is the bioanalysis, second is the bioscreening, bioarchitectures and biosensor. And in all these fields, we have developed some good researches. We have developed some miniaturized devices that can be used for bioanalysis, and we are developing the on-chip bioscreening processes. So on the small chip area, we can screen billions or trillions of biomolecules together. And also we are interested in making the bioarchitectures using the unusual DNA structures. We can use some novel biomaterials for self-assembled autonomous assembly. And here is one of my recent interests. We are developing a biosensor. Just like a strip of paper, this paper strip can be used by combination of different areas to develop the lab-on paper. So today I am going to tell you about something about the lab-on paper that we call the point-of-care nanobiosensor. So this is the ultimate outline of my project. So for early diagnosis, something that can be used at home to get the primary symptoms of our disease or how the disease appear. So by targeting the early diagnosis and to make it for the barrier-free point-of-care biosensor for our healthy aging society. So what we are planning here to integrate three different areas. One is the nanosensor development that we use to improve the sensitivity of signals from the antigens. And second, we are developing our own new biocrops that can be developed for any specific target of diseases. And third is the solution process MEMS device, electronic mechanical devices that can be used to develop suppose like micro pumps. So these micro pumps can be integrated into the paper chip, and we can control the flow of fluids in the paper chips. So by integrating all these three, what we are targeting here to make a novel paper chips looks like here. You can see here that your blood drops or your saliva from your mouth can be integrated, can become in this chip, and then can process through many micro pumps and devices, and ultimately you will get a signals that how many biomarkers in quantitatively how many biomarkers in your blood that can help us to detect the risk or symptoms that we are going to have in near future. So we are trying to develop such type of very common, very affordable chip that can be used by a common man, and that can be used at home actually. So no need to go any healthcare facilities or free instrumentation. So this is one of our target, and basically today I will just introduce you the second part, how we are developing the biocrops. So in one of my major areas, the directed evolution. So how we can use the Darwin concept to generate the new biomolecules in our test tubes. So you know that now we are able to breed any type of animals and crops and golden rice and any type of color of roses. So similarly, how to develop some new breed of biomolecules that can have more function compared to the wild type molecules. So that is one of our target. And once we found such type of super biomolecules, we can integrate or we can implant these bioprobes on the, suppose say, carbon nanotubes that we use as a nano device, and we can analyze some target protein that are really in very low concentration in our blood. So such type of system can help us to detect the disease in early stage. If I give you example of the Alzheimer's disease that appears after 10 years of onset of symptoms. So we must need to find that when such type of mental disorder or some brain disorder appear in our body, then if we can find before the symptoms appears, we can better treat the problems. So our target to make such biosensors that are very sensitive to detect the biomarkers that represent to such type of chronic disease in our body. So little bit tell me you that if you look back the era in 1960, it was led by the chemical era, and then in 1980s we have the molecular era, and in 2000 we have the era of genomics and proteomics, and then in I think coming years now we are going to have the era of peptide drugs, bio drugs that we call. Now you will see that in 1960, this era has been appeared mainly focused on the activity control, how we can control the activity or how we can increase the activity. And in later, now we are coming to control the safety parameter, that how we can make safety of our life. So in 1960, we used the small molecule drugs, which is less than 5 Dalton, like aspirin we use for headache. So these are small molecules that are needed in the early. But in case of small molecules, of course, it can easily absorb by our bioavailabilities, and it has high metabolic stability. But in case of the bigger biomolecules that we use, the antibodies in our body, that is more than 5,000 Dalton. So in case of antibodies or other bigger molecules, it's a very less oral bioavailability and low metabolic activity. But in case of small biomolecules, they are not very specific at all. And in case of antibodies, although they are specific, but again some issue like oral bioavailability and low metabolic activity is the issue. So how about to target here? Not a small, no bigger, but small molecules that can be of some 10 amino acids like 1 kilodalton or something like that. So what we are trying to make a center point of this, and recently these small amino acids or peptide molecules has been highlighted very much in recent years. So you will see that around 1 to 9 amino acids are working as a very potential drug candidate in the coming era of drug discovery. So what we are planning to use as a peptide, small biomolecules as a peptide, and to use as a drug or in the medical or in the diagnostic purposes. So what is our technology that we are using? The directed molecular evolution, and we have developed the cDNA display technology and have published in many papers. And so what is this evolution technology? We have many candidates. So what we do, first we make a library of biomolecules, say DNA molecules of a small length, and we make the mutation in the small molecules, and we create a huge library. Huge means it's a billions or trillions. So 10 to 12 or 10 to 13 or 14 type of biomolecules, and then we screen these biomolecules against a particular target. Suppose if we have a target of hypertension, one of the example is renin. Renin can be as a biomarker for our cardiovascular risk. So if we can have renin here, and we find which biomolecules have a good affinity and can bind with the renin. So once we select those particular candidates that can be binded with these target molecules, and we can select and we can discard other biomolecules. So what we are doing, we are basically screening some good candidates that can be work as a highly functional biomolecules. And again, rotating these cycles, we can in every time around we can make it more functional, more enrichment by making the selection evolutions. So these are our that we call the cDNA display technology. So little bit about cDNA display technology. That is the work as actually how we do in the test tube. We have the various type of biomolecules, And then we link these biomolecules with a linker DNA, and this linker DNA help us to make a covalent linkage between the genotypes and the phenotypes. So that's why we call it amine display, because usually in the in vitro or in vivo expressions, the phenotypes become separated from the genotypes. So we don't know which type of phenotype has the, because to read the phenotype we should know the genetic sequence of the sequence. So here what we are doing, we are making a linkage between the genotype and phenotype using this small covalent link, puromycin linker. And now today I am going to tell you about one of the target, renin. Renin is one of the protein, actually proteas that work to help us to know the status of cardiovascular disease. So what will happen in case of from our liver, the angiotensinogen is released, and then the renin which is from liver is work on angiotensinogen and it convert to angiotensin 1 and then 2, and then it stimulates the cortex, adrenal cortex to release the aldosterone, and then ultimately this aldosterone increase the blood pressure. So in case if you having some problem in blood flow, then this renin can be used to detect the problems. But because this renin is usually in the picogram per milliliter of concentration, so it's very less. So we still don't have such devices that can help to detect the renin in such a low concentration. So what we are challenging here to detect using this method, so first we make the library of biomolecules, and then using this selection and screening, we find the best candidate during this test tube evolution approach. So there are some results that we make this library. It's a small library of around 200 bases, where we have a peptide library, where we have a random amino acids. Random amino acids, in 8 amino acids we have changed all the 20 amino acids type of series. So ultimately using the random you can make the library of 2.5 × 10. So you can make 25 billions of molecules by just making the random library of peptide. And then making the in vitro selections, you can find which is the best candidate in this 100 billion of candidates. So what in the first round of selection, we got some good candidates. There are the 36 clones, and you can see the library, the sequence of all the peptides amino acids, and we have made the homology of the sequence, and we found these 2 clones which are repeated during the selection must be having some good functional against the renin binding. So out of these few candidates, we have selected PI peptide 07, and we found that this peptide has a good binding affinity with the renin in the micromolar region. So there are some other candidates that we found, and among these, then we have evaluated our biomolecules, that how it works. So we have 3 methods here. One is the BICON, that is very famous to know the binding affinity between 2 molecules. So we have evaluated by BICON using the SPR method, and then we also check this method with a very simple method that we call the lateral flow assay, because BICON is very expensive instrument. It's not easy to find anywhere, but such type of method can be easily done in some common laboratories, that we call the lateral flow assay. I will tell you later. And the third method we use to evaluate the peptides by the electrochemical assay using the depth chips. I think today we are going to have one poster presentation about this depth chip, and also about the final presentation about the depth chip. So maybe you can have more knowledge in the later presentation. So this is the result of BICON, that in case of BICON we found that peptide 07 and peptide 22 has good binding affinity against RENs in micromolar range. Although we are expecting to make it nanomolar, but still we are trying to make it more evolved, to make more higher binding. And this is the second approach that we evaluated. So what we do, we use the gold nanoparticles. Gold nanoparticle is a very small particle that is around 40 nanometer in diameter, 40 nanometers. And on these nanoparticles we have attached the renin target, okay, and then we have used these paper strips that we call the nitrocellulose paper strips, and we have spotted our peptide aptamers that we find from the screening. So we have spotted the peptide aptamer here, and we probe that these gold nanoparticles. So there is a one unique property of gold nanoparticles. If they remain separated, they have a color, yellow in color. But when they aggregated each other, okay, the color become violet or colorless. So this is one of the plasma property of gold nanoparticles. So in case of, if there is a good binding between the renin and the aptamers, the color will be changed. So by just visual inspection, we can confirm that whether our renin is binding with the peptide or not. So what we have done, if the gold nanoparticle will go through this strip, and if we meet with the peptide aptamer, then the color will be changed. So by just changing the color, we can know whether our bioprobe has an affinity against the target or not. So there are some results for some few peptides, and you can see we compare 7, 13, 15, 17 and 22 aptamers, and in case of 13 and 17 we got the signals around 56 and 19 as an optical intensity. So out of these 5 aptamers, we could select 13 and 17 as some few bioprobe target molecules against the renin. And next we have evaluated our this probe by the electrochemical method. So what we have done, that is the disposable electrical printed chip. Now using the simple printing method, we can print the electrode on the paper. So it's a very I think cost effective and rapid. So like A4 size, we print the paper. Similarly, we can print the electrode on the paper. So it's a very cost effective method, and just printing the tag on the paper, we immobilize the antibodies and then targeting renin and checking the peptide aptamers, which is immobilized with the gold nanolith. And we could get the signals that in case of 280 microgram per ml, we could get the signals. So with this setup, we also can able to detect the renin at 280 microgram per ml. Although it is not so effective, not so sensitive, so what we are trying to do to make it more to increase the affinity of our bioprobe and to make the detection limit by nanogram or picogram. So this is another some other methods in sandwich assay, because the efficiency was not so good. So we have changed the another method which we call the competitive assay. So what we have done, on the depth surface, on the depth chip, we have the target molecules, and then in the sample, in case of sample, if you have low renin, you will get more signals, but if you have more renin, you will get less samples. So this is the competitive assay between the unknown renin and between the known renin. And using this method, we could able to detect the renin at 280 nanogram per ml. So we could increase the sensitivity of this method by changing the methodology. So till now we could be able to get the new probe for renin, which can be as a biomarker for cardiovascular disease, and we have integrated this biomolecules on the depth chip, and this depth chip can be used to detect the renin concentration maybe in your home. So there is no need to go for any healthcare facilities. If you have that depth chip and if you have this method, you can get the renin concentration in your blood, and you can able to know how risk you are going to have in your cardiovascular related cardiovascular diseases. So this is something which I was to have, because it is something very new, how we can develop new biosensors and we can get early diagnosis of diseases in time. Because such neuro brain order or other disorders, if you know the disease in time, you can better treat the disease. Suppose in case of influenza virus that we have last year, more than 500 people died in Rajasthan. It's very bad actually. So if we have such type of technology and detection method, so we don't need to go anywhere. Even at the home, we can know whether we have normal common cold or whether we have an influenza virus. And once we know, then we can further go for the health checkup. So briefly, high affinity peptide aptamers probes were identified for this renin using our technology. And these aptamer probes can be successfully integrated onto the nano devices and can be able to detect the renin in few hundred nanogram per ml in samples. And a portable electrochemical biosensor platform is designed and demonstrated for point of care applications. So these are one of the new biosensors that we have developed. And yes, this is one of the picture where the work has been done, JAIST. It is a peak summer, and this is the building of JAIST, and here we have the one of the famous mountain, Haksan, around 2000 meter height. And so this is one of the winter, and yes, we also, all the seasons are so beautiful in JAIST, spring and autumn. And I greatly acknowledge the work to my collaborator in Calcutta Laboratories. So thank you very much for listening me.
Thank you for your presentation. So now this paper is open for discussion. Is there any questions or comments from audience?
Yes, please.
Hi, sir. So I want to ask how this technique would be against viruses.
Yes. So like here we use the renin as a target. So if you have a virus, so virus has the nuclear protein and cell membrane protein. So you can target some protein of cell membrane as a target. So if you incubate your library of your probe library with the virus, so some virus, the virus protein can, like in influenza, we have H1N1 protein. So this protein can be as a target for the probe.
Sir, what about the inborn diseases from to fetus by mother? That type of disease, how can we detect this type of?
To fetus to mother?
Mother to fetus, such as some migrainal diseases sort of thing, cardiovascular diseases like hole in heart.
So you mean the genetic disorder? Genetic diseases?
Some sort of genetic also and cardiovascular means you are saying that preliminary we can determine many type of diseases. So how this technique would be helpful for those types?
So I am not actually getting you. Your question is that how we use this technique to get the genetic heredity disorder or?
Yes, sir. Genetic disorder also, and how we can cure those type of diseases also. First determination also, and then curing also.
Well, here we are talking about diagnostic only, not curing actually. Although this can be as a drug candidate also, but this biosensor is working for diagnostics. And rather concerned that you are talking from fetus to mother or mother to fetus, whatever. So, well, if we have some sample from fetus sample, then if we can check or we can compare both the parent and fetal samples, how they differentiate with each other. So, such information can help us to know the difference between the maternal sample. So, yeah, because the point is here that sensitivity, because this system have very highly sensitive. So it can detect the target even in the nanogram to picograms. So even if your sample have very low amount of antigens or difference between the enzymes or some other, so it can be easily detectable like this. So yeah.
Is there any question or comments? If not, it's fine.
Enzymes or some other, so it can be easily detectable.